What if everything humans believe about life, death, and time is only partially true? What if death is not always an ending, but sometimes a pause so long that it feels eternal? These questions once belonged only to philosophers and writers of science fiction. But in the frozen silence of Siberia, science itself was forced to confront them. Beneath layers of ice that have not melted since the last ice age, a tiny living being waited, untouched by history, untouched by time, until modern humans accidentally knocked on its frozen door. When that door opened, it did not just reveal a microscopic creature. It revealed a crack in our understanding of life itself. Siberia is not just cold land. It is a graveyard of time. Vast areas of this region are locked in permafrost, ground that has remained frozen continuously for tens of thousands of years. Unlike ordinary frozen soil, permafrost acts like a natural freezer set permanently below zero. Anything trapped inside it, plants, animals, microbes, can remain preserved for unimaginable lengths of time. Scientists have long known that permafrost holds clues to Earth's past climate, ancient ecosystems, and extinct species. What they did not expect was to find living time capsules still capable of waking up. In the early 21st century, Russian scientists began detailed studies of Siberian permafrost to understand how life adapts to extreme cold. Near the Alazaya River in northeastern Siberia, researchers drilled deep into frozen soil layers that had not thawed since the Pleistocene epoch. These layers formed when mammoths still roamed the earth, and early humans were struggling to survive in harsh climates. The samples were brought to laboratories under sterile conditions to avoid contamination. At first glance, the frozen soil looked lifeless, as expected. There was no movement, no sign of biological activity, only ancient ice and dust. Then the thawing process began. Slowly, carefully, the frozen samples were warmed just enough to melt the ice without damaging any potential biological structures. Under the microscope, scientists observed something extraordinary. A tiny organism began to move. At first, it was subtle, a slight shift, a twitch that could easily be dismissed as an illusion. But the movement continued, becoming deliberate and purposeful. What they were seeing was not a chemical reaction. It was a living organism coming back to life. Further examination identified the organism as a rotifer, a microscopic multicellular animal commonly found in freshwater environments. Rotifers are usually less than 1 millimeter in size, invisible to the naked eye, and completely unknown to most people. Yet this insignificant-looking creature carried within it a biological secret that challenged modern science. Carbon dating of the surrounding soil confirmed that the rotifer had been frozen for approximately 24,000 years. This meant that the organism entered its frozen state during the last ice age and remained there while human civilization evolved from stone tools to smartphones. The research was conducted by scientists from the Institute of Physicochemical and Biological Problems in Soil Science under the Russian Academy of Sciences. One of the lead researchers, Dr. Saint Malavin, later explained that the team initially struggled to accept what they were witnessing. The idea that a multicellular organism could survive frozen for such an immense period went against established biological assumptions. To rule out error, the scientists repeated the experiment multiple times with different samples. Each time, the result was the same. Rotifers revived, moved, fed, and behaved normally. The most shocking confirmation came when the rotifers began to reproduce. Reproduction is one of the strongest indicators of full biological recovery. It proved that their DNA had not degraded, their cellular machinery was intact, and their life cycle could resume as if time had never passed. This was not survival in the usual sense. It was revival after a geological pause. The findings were peer-reviewed and published in the scientific journal Current Biology, making the discovery part of verified scientific record rather than speculation. To understand why this discovery mattered so deeply, one must understand how freezing normally affects living organisms. In most animals, including humans, freezing causes ice crystals to form inside cells. These crystals expand and puncture cell membranes, destroy organelles, and irreversibly damage DNA. The brain is particularly vulnerable. Neurons begin to die within minutes of oxygen loss or freezing, and once neural connections are destroyed, memories and consciousness cannot be restored. This is why human bodies exposed to extreme cold, even if later thawed, do not come back to life. For decades, cryonics organizations have claimed that freezing humans after death could preserve them for future revival. However, no cryonically frozen human has ever been successfully revived. The damage caused by freezing remains far beyond current medical technology to repair. In this context, the rotifer discovery was both inspiring and unsettling. It proved that nature has already solved a problem humans cannot yet overcome. The key to the rotifer's survival lies in a biological state known as cryptobiosis. In cryptobiosis, an organism reduces its metabolic activity to nearly zero. Chemical reactions slow to a crawl, energy consumption drops dramatically, and biological processes essentially shut down. The organism is neither alive in the normal sense nor dead. It exists in a suspended state, protected from environmental damage. Rotifers achieve this by producing special proteins and sugars that stabilize their cells during freezing. These substances prevent ice crystals from forming sharp edges that would rupture cell membranes. They protect DNA and maintain the structural integrity of tissues at a microscopic level. Cryptobiosis was known to exist in some organisms before, including tardigrades, often called water bears, and certain nematodes. These organisms can survive dehydration, radiation, and extreme temperatures. However, the duration of survival observed in the Siberian rotifer was unprecedented. Previous studies documented cryptobiotic survival for years or decades, not tens of thousands of years. This discovery extended the known limits of life far beyond what science previously believed possible. As news of the revival spread through the scientific community, experts began re-evaluating long-held assumptions. If a multicellular organism could preserve itself for 24,000 years without cellular damage, what else might be possible under the right conditions? Scientists emphasized that rotifers are biologically simple compared to humans. Their small size, simple tissues, and unique protective mechanisms make them ideal candidates for cryptobiosis. Humans, by contrast, are incredibly complex organisms with specialized cells and organs that cannot currently survive such extreme suspension. Despite this, researchers see enormous value in studying these organisms. By understanding the molecular mechanisms that protect rotifer cells during freezing, scientists hope to improve cryopreservation techniques for human tissues and organs. One immediate application could be long-term organ storage for transplants. Currently, donated organs have a very limited preservation window, often just a few hours. If organs could be safely frozen and stored for longer periods, it could save countless lives. What if one of the biggest mysteries of science is not buried in space, but frozen beneath the ice of our own planet? Russian scientists revived a microscopic creature after nearly 24,000 years trapped in Siberian permafrost. Something that should have been dead woke up again. It moved. It lived. It survived time itself. But here is the mystery. If nature can pause life for thousands of years and bring it back, could the same happen to humans? Could life be stopped, stored, and restarted? Could humanity one day cheat death? Today, we are going to uncover this unbelievable scientific mystery and explain how this happened, why it shocked scientists, and what it could mean for the future of human life. Before we begin, make sure to like the video, subscribe to the channel, share it, and tell us in the comments where you are watching from. Now, let's enter the mystery. The implications extend beyond medicine. In space exploration, long-duration missions to distant planets may require astronauts to enter some form of suspended animation to survive journeys lasting decades. While this remains science fiction today, discoveries like the rotifer revival suggest that nature has already found ways to pause life safely. The challenge for humans is to learn how to replicate or adapt these mechanisms. At the same time, scientists caution against exaggeration. The revival of a rotifer does not mean humans will soon be frozen and brought back to life. It does not suggest immortality or resurrection. What it does mean is that life is more flexible than we once believed. The boundaries between living, dormant, and dead are not as clear-cut as textbooks once suggested. There is also a philosophical dimension to this discovery. For most of human history, death has been understood as an irreversible transition. Once life ends, it does not return. The rotifer challenges this idea, at least at a biological level. It demonstrates that under certain conditions, life can pause for thousands of years and then resume. This forces us to reconsider what death truly means. Is death always an end? Or is it sometimes a state we do not yet fully understand? 24,000 years ago, when this rotifer entered its frozen sleep, humans could not imagine modern science, technology, or global civilization. Entire species have gone extinct since then. Empires have risen and fallen. Yet this tiny organism remained unchanged, waiting beneath the ice. Its revival is a reminder of how small human history is compared to the timeline of life on Earth. The discovery also raises new questions. How many other organisms are frozen in permafrost, waiting to be discovered? Could ancient bacteria or viruses also be revived? And if so, what risks might they pose? Scientists are already cautious about the possibility of releasing ancient pathogens as permafrost melts due to climate change. The rotifer revival highlights both the promise and the danger hidden beneath the ice. As research continues, scientists around the world are collaborating to study cryptobiotic organisms in greater detail. Advances in molecular biology, genetics, and nanotechnology may one day allow humans to mimic some of these natural protective mechanisms. For now, such possibilities remain distant, but history has shown that today's impossibility often becomes tomorrow's reality. This discovery does not give us answers about human revival, but it gives us something equally powerful: proof that nature has limits are wider than we thought. The rotifer did not defeat time, it simply stepped outside of it. While human civilizations changed and the planet evolved, this tiny creature waited in silence. Its story is not about immortality, but about patience, resilience, and the hidden potential of life. And as scientists continue to study the frozen archives of Earth, one truth becomes clear. The ice of Siberia is not just frozen ground. It is a library of life, filled with stories we are only beginning to read. As scientists absorbed the implications of the rotifer revival, the focus slowly shifted from astonishment to analysis. The initial shock faded, replaced by deeper questions that demanded careful, honest answers. Could this phenomenon ever apply to humans, or was it forever limited to simple organisms? To understand this, researchers began examining not only what made rotifers special, but also what makes humans fragile in comparison. Human life depends on extreme biological complexity. Our bodies are composed of trillions of cells, organized into specialized tissues and organs that work together in precise harmony. The human brain alone contains around 86 billion neurons, each forming thousands of connections. Consciousness, memory, personality, and identity emerge from this delicate network. Unlike rotifers, which can shut down most of their biological processes without consequence, human cells are constantly active. When that activity stops, damage begins almost immediately. Oxygen deprivation kills brain cells within minutes. Freezing accelerates destruction by creating ice crystals that tear through cell membranes and disrupt the fine structure of neural networks. This is why modern medicine cannot revive someone who has been frozen solid. Even if the heart could be restarted, the brain would no longer be the same. The person would not return as they were. From a scientific perspective, death is not just the stopping of the heart. It is the irreversible loss of brain function. Any discussion of human revival must confront this reality. Despite these challenges, the idea of preserving humans at low temperatures has fascinated scientists and futurists for decades. Cryonics emerged in the mid-20th century as a controversial attempt to preserve human bodies after death by freezing them in liquid nitrogen. Supporters of cryonics argue that future technology may one day repair the cellular damage caused by freezing and restore life. Critics point out that there is no evidence this will ever be possible. As of today, cryonics remains a hope, not a proven science. No cryonically preserved human or animal with a complex brain has ever been revived. The rotifer discovery did not validate cryonics, but it did force scientists to reconsider one key assumption: that long-term freezing always destroys life. Nature had provided a counterexample. However, experts were quick to stress that rotifers and humans are separated by an enormous biological gap. Rotifers are tiny, simple organisms with few cell types and no complex nervous system. Their ability to enter cryptobiosis evolved over millions of years as a survival strategy. Humans lack this evolutionary adaptation. Still, the question refused to disappear. If evolution could produce organisms capable of surviving thousands of years in suspended animation, could science one day engineer similar protections for human cells? Researchers began studying the molecular tools rotifers used to survive freezing. Among these tools are specialized proteins that bind to cell membranes, preventing them from collapsing, and sugars that replace water molecules, stabilizing cellular structures during dehydration and freezing. These mechanisms act like a natural form of cryoprotection, something human cells do not naturally possess. Modern cryopreservation already uses artificial cryoprotectants to preserve cells, embryos, and small tissues. Human sperm, eggs, and embryos can be frozen and later thawed with high success rates. Certain tissues can also be preserved for limited periods. The problem arises when scientists attempt to freeze large, complex organs or entire bodies. Cryoprotectants often become toxic at the concentrations needed to prevent ice formation, and uneven cooling can cause catastrophic damage. Researchers now see cryptobiotic organisms as teachers rather than miracles. By understanding how nature avoids these problems, scientists hope to develop safer cryoprotectants and better preservation techniques. Some laboratories are experimenting with vitrification, a process that turns biological material into a glass-like state without forming ice crystals. While vitrification has shown promise for small samples, scaling it up to entire human bodies remains far beyond current capabilities. Beyond medicine, the rotifer discovery has implications for space exploration. Human missions to distant planets or star systems would require travel times far longer than a human lifetime. Suspended animation has long been proposed as a solution, allowing astronauts to sleep through decades-long journeys. Until now, this idea belonged almost entirely to science fiction. But the existence of cryptobiosis in nature suggests that long-term biological suspension is not fundamentally impossible. It is merely something humans have not yet learned to achieve. At the same time, scientists must confront the risks associated with reviving ancient life. As permafrost melts due to climate change, microorganisms that have been frozen for thousands of years are being released back into the environment. Some of these microbes may be harmless, but others could pose risks to modern ecosystems and human health. The revival of a rotifer was controlled and intentional, but nature may soon begin performing similar experiments without human oversight. This adds urgency to the study of ancient organisms and the development of strategies to manage potential threats. Public reaction to the rotifer discovery revealed how deeply it touched human fears and hopes. Some saw it as proof that immortality might one day be possible. Others feared it was the first step toward disturbing forces best left untouched. Scientists emphasized the importance of restraint and honesty. The discovery did not promise eternal life. It did not erase death. What it did was expand the known limits of survival and force humanity to confront how little it truly understands about life. Philosophers and ethicists also entered the conversation. If humans could one day pause their lives and resume them centuries later, what would that mean for identity, responsibility, and society? Would a revived person still belong to the world they left behind? Would they have rights, memories, or obligations? These questions remain hypothetical, but they highlight how deeply the rotifer discovery reaches beyond biology. Looking back through history, it becomes clear that scientific progress often begins with small, unexpected observations. Penicillin was discovered by accident. Electricity was once a curiosity. The rotifer revival may represent a similar moment, a small discovery with the potential to reshape entire fields of knowledge. Whether or not humans ever achieve long-term suspended animation, the study of cryptobiosis will almost certainly influence medicine, biology, and our understanding of life itself. For now, scientists remain cautious. They acknowledge that human revival from deep freezing is not possible with current technology and may never be possible at all. Nature does not guarantee that every solution it evolves can be copied by humans. Some adaptations exist only within very specific biological contexts. The rotifer's ability may be one of those rare gifts perfectly suited to its simple form of life and impossible to translate to complex organisms. Yet science is not built on certainty alone. It advances through curiosity, skepticism, and persistence. As research continues, laboratories around the world are examining extremophiles, organisms that thrive in conditions once thought incompatible with life. Each discovery adds another piece to the puzzle, slowly expanding the map of what life can endure. The rotifer is now part of this growing picture, a symbol of life's ability to wait, endure, and resume when the time is right. In the end, the story of the frozen rotifer is not about humans conquering death. It is about humility. It reminds us that nature has been conducting experiments for billions of years, far longer than humans have practiced science. Sometimes, those experiments reveal answers we did not even know to ask. The rotifer did not wake up to change the world. It woke up because conditions allowed it to do so. But its awakening changed how humans see the boundary between life and death. It showed that life is not always a straight line from birth to death. Sometimes it is a pause, silent, frozen, and patient. Beneath the ice of Siberia, countless secrets may still lie waiting. Some may never awaken. Others may one day emerge to challenge our understanding once again. The revival of a tiny rotifer after 24,000 years is not the end of a mystery. It is the beginning of a much larger one. And the most unsettling truth remains this: if life can wait that long once, there is no guarantee it cannot surprise us again.